In this video, we'll talk about the structure of lipids and the biochemistry of lipids. And we'll also look at some of the stereochemistry of lipids. So a lot of the information that we're learning today, we have learned in biology, but now we're going to go into further detail about some of the chemistry of lipids. First of all, we'll discuss what fatty acids are. So they are carboxylic acids, but they can be quite long. So they can run from 4 to 28 carbons long. Short chain fatty acids are considered to have less than 6 carbons. Medium chain are between 6 and 12. Long chain have 13 to 21. And then very long chain fatty acids have 22 or more carbons in their structure. Most fatty acids have an even number of carbons, and most of them are straight chained. And even though they have a polar carboxyl group, they're non-polar because their chain is so long. So even though there is that presence of a carboxyl group, they're non-polar, meaning that they're hydrophobic. So that's going to impact how they interact in our bodies. So the first type of fatty acid are saturated fatty acids. And when we say saturated, we mean that they're saturated with hydrogen. So in these cases, you only have single bonds between the carbons in the carbon chain. So every single carbon has as many hydrogens as possible combined to it. So there's 3 different representations here. This is the structural formula. This is the space filling model. So if you were building the model from a model kit, and this is the skeletal formula, where every little bend is another carbon. Sometimes if we're feeling lazy, we'll draw it this way. So because they're saturated, it means that they're very linear, they're very straight. There's no bends or kinks in the chain. So that means that they have a strong amount of London forces, which makes them have high melting points, which means that saturated fatty acids are solid at room temperature. Our second example are unsaturated fatty acids. So these have at least one double bond in the fatty acid chain. So this is oleic acid, and this is where the double bond is. So you can see it's not saturated with hydrogen anymore because we've lost 2 to make that double bond. So if we draw it this way, it's still showing the linear form, but actually it causes a kink in the chain if there is that double bond there. So you can see that these are no longer as straight. So because of that, the London forces are weakened because of that inability to stack up as tightly to one another because of those bends. So these ones have lower melting points, which means that they tend to be liquid at room temperature. So triglycerides, that's what we call fats and oils. Those are the things that we consume for energy. So what happens is we have a glycerol molecule, which is a tri-alcohol. So it's 3 carbon molecules with a hydroxyl on every single carbon. So that's glycerol. And then it combines with 3 fatty acids through condensation reactions. So you can see there's a hydrogen coming off of the glycerol and a hydroxyl coming off the fatty acid. And then we end up with a triglyceride and 3 waters because there's 3 condensations that are happening. The bonds are called ester linkages, and that should sound familiar to you because we know that a reaction between alcohol and a carboxylic acid will form an ester. So let's talk about fats and oils. So saturated fats are solid at room temperature and are called fats, and then unsaturated fats are liquid at room temperature and are called oils. And this image really does show how neatly the fatty acids that are lined up in the saturated fat, but how unneatly they're lined up in the unsaturated fatty acid. So our textbook has a helpful hint. So the condensed chemical formulas for some lipids are in section 34 of the data booklet. You don't need to know specific fatty acids, but if you're given the formula, you should be able to predict their differences in melting point based on both the length of the carbon chains and the amount of unsaturation. So if it's saturated, it's going to be solid, and the longer it is, the higher the melting point. And then the more degrees of unsaturation there is, then the lower the melting point. You should also determine the product of a reaction between fatty acids and glycerol. And then don't forget when you're doing that to balance the 3 water molecules that are produced. So we've already discussed all of those things, but that is something you need to be able to do on your chemistry option exam. So one thing we can do to determine how unsaturated a fat is to determine its iodine number. So iodine can undergo addition reactions with double bonds in fatty acids. So it will turn the iodine from brown to colorless. And the iodine number is determined to be the mass of iodine that is absorbed by 100 grams of the sample. And to find the iodine number, you just keep adding iodine until the brown color no longer disappears. So you can see this example where the addition reaction has happened right here. So there were 2 double bonds, and now we have iodines in those positions instead. This table actually is from our textbook, and it shows some examples of some iodine numbers. And you can see that animal-based fatty acids do not have as high an iodine number as plant-based, with a couple of exceptions. So cod liver oil, remember fish have oils or unsaturated fats rather than saturated fats. And one example of a plant oil, coconut oil, is actually quite saturated. And that makes sense because it is solid at room temperature. So you are required to calculate the iodine number, and it's not a difficult calculation at all. It's just the mass of iodine that reacted divided by the mass of the lipid times 100. So fairly simple. So in this example, we see that 11.5 grams of iodine reacted with 18.5 grams of lipid. So we're to calculate the iodine number. So let's calculate that. So our number for iodine is equal to the mass of iodine that reacted over the mass of the lipid times 100. So that is equivalent to 11.5 grams divided by 18.5 grams times 100. So we get an iodine number of 62.2. We can also determine the number of double bonds if we know the iodine number, the molar mass of the lipid, and then some stoichiometry thrown in for some fun. So basically you have your unsaturated lipid times the number of moles of iodine will tell you how much the saturated lipid is. So there are a few steps that we need to do for this one. So here's an example, and then we'll work through it. A long chain carboxylic acid has a molar mass of 302.45 grams per mole and an iodine number of 420. So we want to calculate the number of carbon-carbon double bonds in a molecule of the acid. So the first thing that we need to do is assume we have 1 gram of lipid to calculate the mass of iodine that has reacted. Then we can calculate the moles of iodine. Then we can calculate the moles of the lipid. And then we can find the mole ratio of lipid to iodine. So we just divide the moles by the lowest mole. So the first thing that we need to do is assume that 1 gram of the sample lipids to calculate the mass of the iodine reacted. So the iodine number is equal to the mass of iodine over the mass of lipid times 100. So we can then figure out the mass of iodine by substituting all our numbers in. So we know that the 420 is equal to the iodine number, equals, again, the mass of iodine to 1 gram of lipid times 100. So if we divide 420 by 100, that will give you the mass of iodine in 1 gram. So the mass of iodine is 4.20 grams. And that's iodine as a 2. So now we need to figure out how many moles of iodine we have. So remember, mass divided by molar mass will give us our moles. So the molar mass of iodine, so we would look that up. So we have 4.20 grams of iodine divided by 253.8 grams per mole. So that's going to give us a number of 0.0165 moles of iodine. So we're going back to our old mole relationships here. Our next step is to find out how many moles of the lipid we had. So remember, we had 1 gram of the lipid. So that's our mass. And we were told in the initial problem that the molar mass of the lipid is 302.45 grams per mole. So that is going to give us a mole of 0.00331 moles of the lipid. So now we're done that part. So now all we have to do is to find the mole ratio of lipid to iodine. So we do the mass over the lowest, or the moles over the lowest moles. So the higher mole value was the 0.0165 moles of iodine. And our lower mole amount was 0.00331 moles of lipid. And we end up with an answer of 4.99 moles of iodine for every 1.00 mole of lipid, or more simply, 5 to 1. So there are 5 carbon-carbon double bonds. In this sample. So this isn't overly complicated, it just takes a few steps. So for these ones, the first thing you need to do is find the mass of iodine per gram of lipids. So you just assume you have 1 gram of lipid. So you just take the iodine number and divide by 100. Actually, is all you have to do for that part. So 4.2 grams of iodine was our first part. Then you find the moles of iodine by dividing by the molar mass, that mass. And then, and remember, iodine is 2 iodines. It's a diatomic molecule, so don't forget to do that part. And then find the moles of the lipid. Remember, we assumed we had 1 gram, so then we get that. And then you just take your higher mole and divide your smaller mole, and then you end up with your number of double bonds. Phospholipids are familiar to us as well. Remember, phospholipids are glycerol, 2 fatty acids, and then a phosphate. With this, we didn't talk about this, but the phosphate is now an ionic region, and then we have a nonpolar. So we said that this was hydrophilic and that was hydrophobic in our lessons in biology. But remember, this is ionic, and it is nonpolar. So fatty acids are hydrophobic, phosphate is hydrophilic. When phosphates interact with water, the hydrophobic tails do not want to interact at all. So there are 2 things that happen. The phosphate head forms ion dipole interactions. So the ionic portion will interact with the dipoles in water. And then the fatty acid tails will try to hide themselves from water. So they can either form micelles, which are spheres, like here we can only see a cross-section of that, or they'll form phospholipid bilayers. Now we've seen the phospholipid bilayer in biology when we talked about any of the membranes in our cells. So micelles often form around other nonpolar materials. So vitamins are examples. So when we do our lesson on vitamins, we'll learn that several vitamins are fat soluble because they too are nonpolar. So they're going to be carried around through the blood and stored in tissues within micelles. So that lets them get transported through the bloodstream in micelles. So nonpolar things will be transported by way of micelles. Our phospholipid bilayer, as I talked about already, make up membranes in the cell. They will also have embedded structures, so proteins and molecules that act as markers. So some of them might be channels for transport. Some of them may be markers. They may be used for communication. There's lots of purposes for things to be embedded into the bilayer. So we talked briefly in biology about steroids. We'll talk a little bit more in depth now. So steroids make up our hormones, and they're also a component of the cell membrane. This detail we didn't know. So there's 17 carbons in a steroid, and they're found in 4 fused rings, and that makes up the backbone. And then there's different functional groups that give each of them their unique properties. So the structure on the right is the general steroid form. So there's 1, 2, 3, 4 rings, as I talked about. The one on the right has the functional groups attached. So this molecule here is actually testosterone. So the properties of steroids. The backbone itself is nonpolar because there's no polar functional groups. But a lot of steroid hormones are polar because they do have functional groups that make them polar, and that allows them to be transported more easily through the blood. The ones that don't have polar functional groups have to be carried by carrier proteins that will allow them to get transported through the blood. In biology, we did learn that cholesterol is an important part of the cell membrane. It adds rigidity to the membrane, and that can vary depending on the amount of cholesterol that's in the membrane. So it helps the membrane maintain its shape, and it adjusts and allows the fluidity to be altered. Now, sometimes we have to take steroids for medicinal purposes. Like if you have asthma, you may take a steroid inhaler to help open up your airways. But some people take them for non-medical reasons. And increased steroid use can actually raise your cholesterol levels and lead to an increased risk of cardiovascular disease. So there's always a fine balance of how much of a steroid you take, even for medicinal purposes, to prevent any negative health effects. So one example of a steroid use that's used for not good purposes is testosterone. It's used to build muscle, and it does enhance athletic performance, but it can lead to hair loss and decreased fertility, which are not necessarily characteristics you want to exhibit. So why do we need fat in the first place? Well, one, it's an excellent source of energy. It provides 2 times the amount of energy that carbohydrates and proteins do per gram. Fat is also used to make other lipids that we need, such as steroids and even phospholipids. But we have to eat them in moderation because too much ingestion of fats can actually lead to some negative health impacts. So saturated fats, if we eat too much of those in our diets, they can actually impact our cholesterol levels and lead to an increased risk of heart disease. We don't want that. Unsaturated fats, they're usually formed, if they're in their natural form, they're found in their cis transformation or cis formation. They don't contribute to increased heart health risk. But trans fats do, because the double bond is now in the trans configuration. So they do raise our blood cholesterol and our heart disease risk. This graph is from our textbook, and it shows you the melting points of the 3. So saturated fats have the highest melting point, trans fats are in between, and then cis fats have a much lower melting point, but you can see even for all three, as the number of carbons increases, the melting point also increases. So even though unsaturated fats have low melting points, they still do raise as the length of the chain gets longer. So let's talk about cholesterol. Cholesterol is nonpolar, so it has to be carried through the blood by a carrier protein called a lipoprotein, and that makes the cholesterol more soluble in water. And there's two different types of lipoprotein. High-density lipoprotein absorbs blood cholesterol and takes it to the liver to be eliminated from the body. This is a good thing. So your HDL cholesterol, that's your good cholesterol. So the higher that is, the more you're eliminating your excess cholesterol from your body. Your low-density lipoprotein, on the other hand, actually will deposit cholesterol into your blood, which can form plaque, and that is a bad thing. So you can see there's a difference there. So in the high-density lipoprotein, you have more protein and less cholesterol. So high-density lipoprotein. Low-density lipoprotein, you have only in this example, there's only the one represented and a whole bunch of cholesterol. So this one deposits it in your blood, and this one eliminates it from your blood. So, like I said before, LDL lipoproteins or low-density lipoproteins deposit cholesterol into your arteries. So this is your open artery, and slowly over time, it's depositing more and more so that it's almost blocked. And that can lead to heart attacks and strokes because the blood can't get through, and that's a problem. Our last concept we're going to talk about is rancidity. So fats and oils can spoil, so we have to store them properly. And it undergoes something called hydrolytic rancidity. So the triglyceride undergoes hydrolysis and gets converted to glycerol and fatty acids. So dairy products are prone to this type of spoiling because they're found in water. So that's hydrolytic rancidity. Oxidative rancidity is when the double bonds in unsaturated fats react with oxygen, and that gets catalyzed by light, meaning that oils that are prone to this type of spoiling tend to be stored in darker glass bottles away from light. So you have to store them carefully. Sometimes people will even refrigerate their oils so that you can slow that process down, and your fridge is dark. However, trans fats, how do we get them? So they can be formed during food processing when unsaturated fats are partially hydrogenated, and they undergo reduction reactions with a nickel catalyst. And solid margarines and vegetable oil shortening are made from liquid vegetable oils. And then why do we make them in the first place? Because we know that they're bad for us. Well, originally they didn't think they were bad for us. They thought that because they were still unsaturated, they would carry the properties of unsaturated fats. It took many, many decades to realize that they actually are maybe even more harmful than saturated fats than we thought. But the reason why solid fats have a better flavor and mouthfeel than liquid oils, they give food a different texture and flavor, and trans fats are less likely to become rancid because the cis double bonds are more reactive than the trans double bonds. So trans fats actually increase shelf life and prevent food from spoiling so quickly. Unsaturated fats spoil much more quickly because the fats within go rancid. So that's why trans fats are used.